Welcome to our webinar this afternoon. Excuse me. Before we start, some housekeeping. If you have any technical issues, please send a short message in the question box on the right-hand side of your screen. During the webinar, RSSL would like to invite you to ask questions using the question box also in the right-hand side. At the end of the webinar, time permitting, we will try and answer as many of these as possible. Excuse me. Any we don't get around to, we will answer offline and share them with you along with the link to the recording. Today's webinar is on, the final phase of disinfectant efficiency, implementing a field trial. The speaker is the eminent Dr. Tim Sandle, and I am Nikki Boyle from RSSL. So, my name is Nikki Boyle. I'm a sterile and non-sterile microbiology lead at RSSL. I've recently joined the commercial team here at RSSL. I bring with me over 20 years experience within the pharmaceutical and life science sector. My role is to support clients that require a variety of microbial testing, from routine MLT to more complex EM or sterility type tests. So those are my contact details. So should you need any support, then please reach out. So a little bit about the organisation. For those of you who know RSL, who are we? Or sorry, for those of you who don't know, who are we? So RSSL is a global CRO supporting clients with a wide variety of analytical chemical services. We've been around for over 35 years and are exclusively based in the Reading area on 4 separate sites. So that, for those who don't know where Reading is, it's about 40 minutes west of London and Heathrow. For all 4 sites accommodate over 350 scientists working and supporting clients in 12 different technical areas of microbiology, of which microbiology is just one of them. We support clients globally, both within Europe, the US, and APAC. As such, RSSL are a trusted CRO with a big emphasis on quality, science, and service. This can be shown by our high Net Promoter Score. Because our sites sit close to one another, we can show great agility to manage complex multidisciplinary projects. We also have a dedicated multi-lab project manager and client liaison managers to ensure that the most complex of projects are kept on track and managed well. You may not be aware, but we're also backed by a multi-million-dollar parent company called Mondelez, which allows us to have access to funding and support to expand and grow as markets grow and diverge. So, the technical areas which we specialise in allow us to support a diverse range of biological, microbiological and chemical, physical, analytical services across all phases of clinical development through to commercialisation. We're exceedingly well positioned to support projects utilising our state-of-the-art equipment and experienced technical teams who can advise and support you throughout the manufacturing process. Be that routine material testing, investigative support, or any issues that you may face, such as impurities, foreign particles in your processes. We have a well-equipped and extensive microscopy department which can help build specific particle databases for your manufacturing process. Our biological support is also very extensive, where we can support all aspects of Annex 1 compliance, including microbial identification of any EM isolates or unfortunate positives from your bioburden analysis, using the state-of-the-art MALDI-TOF system. We can also offer a range of turnaround times to ensure you get those IDs through as soon as possible to help support your decisions on next steps. So I think that's enough from me. I would now like to introduce you to our guest speaker today, Mr. Tim Sandle. I'm sure many of you have heard of him and subscribed to his many publications. Mr. Tim has over 30 years experience in the microbiology area and research, pharmaceutical processing. Tim is a member of several editorial boards, and he has written over 900 book chapters, peer-reviewed papers and technical articles relating to microbiology. Tim works as at a pharmaceutical manufacturer within the UK and also finds time to be an associate professor at both the University of Manchester and UCL. So over to you, Tim.
Okay, thank you very much. We'll just do a quick screen switch. Okay, brilliant. Well, it's great to be back with everyone, and especially those who listened to the webinar last month, because in some senses this is a continuation. But if you didn't, then it also picks up on the core subject, which is the field trial. So we'll just do a very quick recap in a second on the different phases of disinfectant qualification, and then we'll move into how to define a field trial, what the key factors are that we may wish to consider, the process of designing the field trial, and then we'll have a look at some real life data in the form of a case study. And then we'll look at some of the things that can go wrong and what we may wish to do about it, and also kind of conclude with perhaps when we may wish to repeat a field trial or other variables that at least make us consider our earlier qualification study. And hopefully this will be useful, because although the disinfectant standards up until the field trial are provided in a reasonable amount of detail, be they European or US standards, the field trial still remains the area that there's no official standard for, yet a number of compendia and regulatory guidances require us to prove that the disinfectants and detergents that we wish to use in real life situations are proven to be suitable in such practical settings. So just as a quick recap, the qualifications required for disinfectants are presented in 3 phases. There are suspension tests, which are those tests normally undertaken by the disinfectant manufacturer rather than the user. As we looked at last month, these are not of huge value beyond an initial sort of screening of the appropriate concentration of a disinfectant. More meaningfully are the surface testing, where a range of different surfaces that we would find in clean rooms or healthcare facilities are screened against individual microorganisms, and these provide a more robust challenge than the suspension test. But all of this is theoretical, it's all laboratory based, and what really matters are how our staff who are employed to undertake cleaning and disinfection actually using the different agents. What are the challenges? How often should we rotate between different biocides? What frequency should we be cleaning and disinfecting overall? And these are the questions that the field trial can provide answers to. And it can also help us to understand when and where we need to conduct environmental monitoring, and also to help us to make sense of the data that we generate. But it's also important, before we jump in, to consider what makes disinfectants effective. So when we're undertaking sort of earlier disinfectant studies, we need to make sure that the microorganisms that we have used in those studies are reflective of those commonly found within our facility, be that pharmaceutical facility or healthcare facility. Because there's no point launching into the field trial if we have missed a trick of understanding our own microbial flora, because the disinfectant may prove to be ineffective. We also need to understand what kind of logarithmic kills we wish to achieve, and how reflective that is of our actual in-use settings. We need to know that our disinfectants are indeed effective across the range of surfaces that, say, we would find in a clean room. So there's little value having just only done stainless steel if we don't understand what the impact might be upon vinyl, because we're going to get different types of microbial adhesion expressed by different species of organisms. And also the earlier testing allows us to go in with the concentration that we think is going to be most effective, and also in terms of the application time, so the time that the disinfectant is in contact with the surface for, and allowing it to be able to traverse and reach the microbial cell. And also because the pharmaceutical facility will undoubtedly contain traces of residues, because cleaning can only be effective to a given degree, it's also important to have understood how our disinfectant is going to react if there are traces of other substances, potentially interfering factors or soiling. So of those points I've mentioned, probably the most important one is ensuring that this suitable range of surfaces have been pre-assessed. Now, it's not necessary to do every single surface, but it is important to understand and have knowledge of all the different surfaces, and then to have undertaken a defensible approach of grouping different surfaces together and deciding which ones are likely to be representative. So in the previous webinar, I spoke, for example, about 2 different grades of stainless steel, and there I think I recommended that the surface steel that's actually got a slightly rougher surface, say 304L stainless steel compared to 316L stainless steel, is probably a better surface to use in a qualification study, because the possibility of microbial adherence is higher. But it's those sorts of things that we need to weigh up, and on the slide are the types of surfaces that we might wish to consider, and have an understanding that how our disinfectants are likely to react with those surfaces, and what the different types of physicochemical interactions between different microorganisms on those surfaces are likely to be, and then to come up with some optimal concentration and contact times that we can deploy. So with all of that established, we enter into the world of the field trial. So the objective of a field trial is to demonstrate that either, we haven't done a field trial before, so we wish to demonstrate how good our disinfectants are, or if we are changing a disinfectant, if we're introducing a new disinfectant, so let's say we're switching from a chlorine-based compound to a peroxide or something, we want to know whether this new disinfectant is better than or equally as good as controlling the bioburden within our facility. And that would be in comparison with an existing product, or as I said, if no field trial has previously been run, then it gives us that first sort of baseline data. And the requirement for a field trial is mentioned in the European and ISO biocontamination standard. It's mentioned in USP chapter 1072, and it's also an Annex 1 requirement. So on many levels, the field trial is the most important qualification test, because it is testing disinfectants in combination with detergents and disinfectants in combination with each other at the point of use, and also looks at the different formats in which disinfectants are supplied. So we can evaluate ready-to-made solutions, or using concentrates and making those up in water, and also the different application techniques. So for example, triple bucket system versus pre-saturated wipe. So we're taking the laboratory testing and the the parameters that we've established within the laboratory, which at this point remain theoretical because we haven't tested those in a real life setting, particularly concentration and contact time. And the field trial, again, unlike the laboratory testing, is not looking at a single disinfectant. It's looking at the 2 disinfectants that we'll be using in rotation, or if we have a sporicide as a less frequent application, it could be the combination of 3 disinfectants, also detergent, also application techniques, as I mentioned. And we can also use that to fine-tune the rotation. We can get an understanding of the spectrum of activity. We can get verification that we have selected disinfectants with different modes of action that are indeed suitably different to give us a wide range of microbial kill. And we can also partly address the issue of residues. So certainly if we're using a disinfectant that's not classed as a low residue disinfectant, we will need to remove residues. Or, for example, if we're using a chlorine dioxide, then we would need to wipe away after the contact time has elapsed, otherwise we will get reaction, say with stainless steel. But even with low residue disinfectants, it's still good practice to at least understand when we need to remove residues, such as changing between 2 different disinfectants. Okay, so with the design process of the field trial, it's important to begin the process with a change control, because we are introducing, say, a novel disinfectant into the facility, and to have a validation protocol in place before the study begins that outlines the length of the study, the number of samples, and the acceptance criteria, because this is like any other form of validation study. And depending on the extent of the facility, how large it is, it's important to consider clean rooms of different types and different activities. So if we have areas that are slightly wetter, there might be water sources, changing rooms, we're going to have corridors, process rooms, and so on. The one area where it's probably less meaningful is something like grade A. So we may want to do a small amount of testing in grade A, but grade A itself is not really going to generate any data, because we're expecting, for the most part, no microorganisms to be present. So generating lots and lots of null data doesn't allow for a particularly meaningful comparison. It's also important that we build into the design a sufficient number of monitoring sessions that allow us to assess the rotational frequencies. So if we're rotating between 2 different disinfectants every week, then we will capture those rotational sessions more quickly than if we are rotating once a month. So how long a field trial runs for depends upon what window of activity and level of monitoring we need to undertake in order to capture a sufficient number of disinfectant rotations. And I would think that 3 is probably the minimum to give us a degree of confidence that the results we obtain are not simply due to chance, but are reflective of the activity of the disinfectant. In terms of the sample types to target with environmental monitoring, then there is little value in taking air samples because our focus is whether the application of a disinfectant on that range of surfaces that we've discussed is effective. So swabs and predominantly contact plates are the optimal samples to take. It's also important as part of the design process that we perform a risk assessment because we want to have an understanding of the level of risk from undertaking the field trial, because say, for example, what would happen if our results are not very good and we see an increase of contamination, or what might happen if we get a bad reaction between a disinfectant and a given surface. And we also need to decide on not only the sort of number of samples that we wish to take within a clean room that gives us a representative amount of data, but also to ensure that we are targeting samples on the different surface types. And also we're doing a kind of combination of floor samples and working height samples. And depending upon the nature of the surface, that will dictate whether the contact plate or the swab is the most appropriate method. Generally, if we can use a contact plate, it does tend to give higher recovery than many swab types. It's also important that we have done some pre-assessment work, which should have come out from the surface studies in terms of the neutralizer that will be added to the culture media. So we need to make sure that our culture media contains a neutralizer that is proven to be capable of neutralizing any disinfectant residues. And this is important even if we're using a disinfectant classified as low residue, because low residue doesn't mean zero residue. And we want to be sure that the results that we're obtaining are reflective of the true microbial population. And ideally, we want to be using a neutralizer that is effective against all of the cleaning and disinfection agents. Otherwise, we might have to use two different types of contact plates, for example, where we've used different disinfectants. So the aim should be to have a universal neutralizer that is suitable, that the medium manufacturer has some supporting data for, that it forms a suitable complex within the agar, and one that is reflective of our surface studies. Also, to the design process, we need to consider whether the detergent we're using is compatible with the disinfectants, because if it isn't, then that will cause us problems down the line. And an example of that could be that if our disinfectant is required to operate along a particular pH range, but the presence of detergent residues alters that pH range, then we might be using a disinfectant that doesn't have the same potency that our laboratory studies might have suggested. I've mentioned residues, and we need to decide up front whether or not we're having any kind of rinsing steps or other processes to remove residues, and then whether we're going to be doing that after detergent, or after each disinfectant application, or just between the switchover of disinfectants. So there's some factors to consider there. In terms of residues, this can either be approached on the basis of a visual inspection of the surface, or we could go for more sophisticated approaches such as surface conductivity testing. We also need to have decided the formats for our disinfectants, because we don't want to introduce a different format later down the line after we've completed the field trial. We want to try and capture those variety of format uses within the field trial. So are we going to be using a concentrate that we will be diluting to put into a bucket? Will we be using pre-saturated wipes or pre-saturated mop heads? Will we be applying the disinfectant in a trigger spray with a standard wipe? Are we going to be using any disinfectant that we need to pass through a sterilizing grade filter? What type of water and what's our water source that we're going to be using? And we also need to define how much disinfectant we'll be applying in a given area, so how much surface coverage, and this is particularly important. We'll be using double or triple bucket systems. So how many clean rooms of a given size does one combination cover, and for how long, if we're preparing a disinfectant concentrate, do we expect to be using that disinfectant for? So in other words, we make up disinfectant in our bucket, we decide on double or triple bucket system. The moment we've added the concentrate to the water, are we going to say we're going to change that every 2 hours or 4 hours? And are we going to use that for one of our clean rooms or for 3 of our clean rooms, et cetera, et cetera. So we need to build all that into the surface study, into the field trial rather, so that the field trial is very, very tight in its representativeness when we use the disinfectant as part of our routine contamination control. It's also important that when we perform the environmental monitoring, that we're undertaking the environmental monitoring under representative conditions. So we want to be targeting clean rooms that have the typical number of personnel present, equipment running, and so on. And that's in order to make sure that the bioburden that we're trying to evaluate these disinfectants against are reflective of what is actually there. So if we were just targeting clean rooms with low use or in unoccupied states, then it's a little bit meaningless because we're not robustly challenging that disinfectant. So again, that's an important part of the design process, as is the timing that we're taking environmental monitoring samples. So we want to kind of get a comparison of before and after the application of a given disinfectant. But ideally, the sort of before measure, we want to be sort of doing almost as near to the longest point between the last disinfection. So we're sampling the surface under the worst case set of conditions. Okay, so I'm now going to try and tie these kind of more abstract notions together in the form of a case study. And this is a real-life field trial that was undertaken to assess the microbial status of clean room surfaces, and it was looking at surfaces just prior to and after the application of disinfectants. And this was evaluating 2 disinfectants. One was a sporicide, which was a hydrogen peroxide, and the other was a non-sporicide, but of a disinfectant with a reasonably wide spectrum of activity. It was a 5th generation quaternary ammonium compound. And the study was run across 3 different classes of clean room based on the EU GMP grading system. And the study was looking at microbial bioburden in terms of the populations, but it was also looking at types of species as well. And it was also considering any differences between rooms of different types and between sampling locations. And the different classes of clean rooms were disinfected according to different regimes, but the sort of standard regime was 4 weeks of the non-sporicide quaternary ammonium compound, followed by 1 week of the sporicide, and then that went into a repeating pattern, so sort of like a 4-to-1 ratio of quaternary to peroxide. And this particular study examined this across 3 rotational cycles, which meant that the study lasted for 15 weeks. So it's a 15-week field trial. And there's always choices to make here, as I mentioned earlier, between how long do you let the field trial run for, but I think you need these 3 rotational patterns to strike that balance between collating a data set that's large enough and one that is representative, but equally something that's manageable, doesn't go on forever. And within this rotational pattern, each clean room was disinfected once a day for the grade C and D clean rooms, and there's also grade B clean rooms included, and these were disinfected twice a day to reflect the greater criticality of those areas. And the concentration and contact times for these disinfectants had previously been assessed through laboratory studies. The method of application was on the floors with a non-shedding mop using a triple bucket system, and for surfaces this was using pre-saturated wipes with a wipe typically folded 4 times, but only one surface of the wipe being used for any one wiping motion. And the different clean rooms were sampled using contact plates, and these contained tryptone agar, and they contained a neutralizer that had been shown to be effective against the 2 different types of disinfectants. And this was a neutralizer that was a combination of lecithin, twin 80 or polysorbate 80, sodium thiosulfate, and L-histidine. The sampling was undertaken where contact plates were used using a contact plate applicator that allows a level of control over the amount of pressure applied, and the timing, the time that the contact plate was in contact with the surface was controlled at 10 seconds. And the plates were subjected to a dual incubation regime, and this was 3 days at 20 to 25 degrees Celsius, followed by 2 days at 30 to 35 degrees Celsius. And this was an incubation regime that had previously been verified as optimal for the typical organisms found within the clean room, and was designed to recover both fungi, which is what the lower temperature predominates towards, and of bacteria. And the rooms were examined under the following conditions. So 6 contact plates were taken in each room, and the timing was as close to the next scheduled application of a disinfectant as possible. And then a further 6 contact plates were taken in each room after the disinfection had taken place, just with an indication of time, so that making sure that surfaces were not wet but had dried, and then environmental monitoring was permitted. So we can call these condition 1 and condition 2 for the later comparison. And samples were taken from different locations, so floor samples predominantly vinyl because the clean rooms were extensively vinyled. Working height samples were predominantly on stainless steel, and other samples, what you might call slightly higher level samples, were taken from acrylic and from glass. So a reasonable representation of different surface types were captured in the study. In terms of the acceptance criteria, the recommended maximal levels from EU GMP Annex 1 were taken into consideration, so we didn't want any clean room recording levels really above these. So that's 5 colony forming units per 25 square centimeters for grade B, 25 for grade C, and 50 for grade D. And the study included an assessment of the differences in microbial counts between the pre and post disinfection condition. And what a successful study would show, so as part of that kind of pre-consideration in the study protocol, was that there would be ideally reductions between pre and post disinfection sessions, or at least no increase in contamination. Ideally, there would be reductions over the course of the study. So we're introducing new disinfectant. Ideally, we'd like it to be better than the previous disinfectant, or at least no worse. We'd want to know if there's any differences between the different grades of clean rooms. And also, as well as microbial numbers, as I mentioned before, would also be interested in the species, because even if we saw reductions in microbial populations, we wouldn't want to see an increase in spore forming organisms, because that might infer that our rotation with our sporicide was ineffective. So I've got some data summaries in a moment, and the results have been divided for each clean room. So the graphs that will be coming up on the next slide display the variation between the pre and post disinfection counts across time, and also to help to illustrate the charts, there's a 2-point moving average, which is tracking the pre and post disinfection bioburden counts. And with these summary charts across the clean rooms, what I've done is use the same scale, so it becomes easier to see what kind of levels of contamination there are between the different clean room grades. So here we have a graph for the grade D clean rooms. So for illustrative purposes, the different clean rooms have been combined just in terms of the presentation for this webinar. So the data shows a reduction in microbial counts following the application of disinfectants across these 3 rotational cycles. And the typical reduction in contamination when the quaternary ammonium compound was applied was 71%. The reduction when the peroxide was applied was 56%, and the overall reduction was 70%. And there were 5 occasions when environmental monitoring results were on the higher side, and these are all confined to the pre-disinfection condition. And in every case, the counts obtained for the post-application condition were considerably lower in value. And then similarly for grade C, the application of the quaternary ammonium compound gave a 63% reduction. The application of the peroxide gave a 77% reduction, and our overall reduction was 66%, and there were no excursions beyond EU GMP action levels. For grade B, the counts are obviously very, very low, but we can still express these in terms of microbial reductions. So for the quaternary ammonium compound, it was 74%. For the peroxide, it was 94%, and our overall reduction was 79%. And there were 4 occasions where environmental monitoring results were high for the pre-disinfection condition, but there were no instances of high counts for the post-disinfection condition. So each clean room showed an overall reduction in microbial counts, and this slide here is all to scale. So you can see obviously we're getting higher microbial counts in grade D. Then we're getting sort of next level is grade C, and then lower counts in grade B. And the parts of the graph that are shaded grey are before the next application of a disinfectant, the precondition. The bars in sort of greeny colour are the post conditions. So you can see there that grade D counts were averaging at just over 16 CFU. After the application of a disinfectant, they dropped to around 5 CFU. Grade C, averaging at 7, drops down to just over 2, and grade B of being under 2 drops down to close to 0. So this allowed a conclusion to be reached that this particular field study was successful, and there was further statistical analysis, tests for significance and so on that were performed to really demonstrate this against the data that was used. And it also gave an understanding of how clean room grades differ, of whether the frequency between sporicide and non-sporicide and the overall frequency of applying any given disinfectant was suitable. We can also take the data, because we've carried out a huge amount of environmental monitoring, and we can use the data to help also consider what might be useful in terms of alert and action levels as well. So there's another application, another sort of use of the data that we can make. And we can also study the data patterns to gain an understanding of how effective is our bioburden reduction through the facility, whether we get build up in certain areas relating to different processes, or whether we're getting higher levels in where we've got clean rooms of different grades in those sort of adjacent rooms. So where Annex 1 is sort of signalling a holistic approach to contamination control, then some of this kind of intensive monitoring can be particularly useful. And I mentioned, as well as microbial counts, it's useful to carry out a level of microbial identification. And obviously a balance needs to be struck between how many of the recovered organisms are identified, but screening a representative number at least gives us an idea of the level of organisms that might show a degree of resistance against certain types of biocides are existing. So particularly the spore forming bacteria and with fungi. So this allows us to sort of work out the appropriate frequency of sporicidal application. So it's that frequency between disinfectants and also the overall rate of application. And in this particular study, a sort of standard phenotypic automated system was used. This happened to be Biolog, Omnilog, but could use MALDI-TOF or a similar system. And the data here in this case did show that there were a occasional low level of spore-forming microorganisms, principally bacillus species of bacteria. In the case of fungi, there was a few cases of aspergillus in the grade D clean room. In each case, these were from the pre-disinfection application and not from the post application. So in this particular case study, the rotation between the quat and the hydrogen peroxide was deemed to be sufficient. So that's a case study for a field trial. There are different factors that can affect the performance of the field trial and of the disinfectants. So the first thing is, the range of different surfaces. What is a suitable grouping of surfaces? So there are some factors we can weigh up here. So we can look at the hydrophobicity, surface roughness, potential for chemical interactions on the surface, how common a particular surface is, contamination risks. So horizontal surfaces will pick up a greater deposition of contamination than slanted surfaces, for example, how close surfaces are to product. So we can use that type of logic to, again, make sure that our sampling is appropriately targeted. We also need to consider the temperature of rooms as well. So one thing I said in the previous webinar was that disinfectant efficacy testing in the laboratory tends to target 20 degrees Celsius, which is fine if we're applying all of our disinfectants in practice to ambient rooms. But if we have cold rooms and so on, we need to make sure that we're including those in the field trial. And that's because each 10-degree drop in temperature makes the contact time double. So we need to kind of test what happens when we apply disinfectants into cold rooms. So it also needs to be considered if that is appropriate. The neutralizer, which I've mentioned before, is important. So ideally we would have established this in the surface testing, and we're simply saying, okay, is the disinfectant we're using in our contact plates the right one for our study? And the early laboratory testing will demonstrate, one, does it actually neutralize the disinfectant, but two, we also need to make sure that it's not toxic to the microorganisms, because that will give us false negative. We also need to consider the rate of drying as well. So again, one thing I mentioned last time was that the laboratory studies might show that we've got a contact time of 5 minutes or 10 minutes is appropriate. But if we're applying our disinfectant in a real life clean room and it's drying in 3 minutes, then we'll need to consider whether we need a additional application or find some way to show efficacy with a reduced contact time. So we need to sort of understand that. Now, there is obviously a degree of residual activity when a disinfectant dries, but it's never going to be as potent as when the surface is wet. And we also need to understand that our disinfectants work in where there's going to be residues of soil. So doing that up front, proving disinfectants work under clean and so-called dirty conditions is important, because once we apply the disinfectant to the field, then we are bound to have different residues. So we need to ensure we've chosen a worst case soiling substance. The European standards recommend bovine serum albumin, and that may or may not be the most appropriate material to use. It would depend upon what's likely to be found in your facility. And then finally, sometimes field trials just don't work. We don't get the data that we want. So we would need to then pause and say, well, okay, is it to do with our application methods? Is it due to our frequency of rotation between sporicide and non-sporicide? Is it our overall frequencies? Did we not quite get the contact time right? Or did we not quite get the concentration right? Or is it just simply that one of our chosen disinfectants is actually not very good? So at the end of the day, hopefully we have established appropriate frequencies and rotations, and we are using ideal disinfectants that are effective against all the types of organisms we might encounter in our clean rooms, that can reduce our microbial populations to a suitable low level, which are compatible with our surfaces, and ideally we have methods that will not be leaving residues. And to do so puts us in a state where we can present good data to a regulator, and this would be something to satisfy under EU GMP Annex 1 and also FDA requirements. So this brings the this part of the webinar to a close. So we've looked at we've done a recap of laboratory testing, we've defined the field trial, we've looked at some of the important factors, we've looked at some of the elements of the design process, we've looked at some case study data, we've considered what affects performance, we've looked at what to do if things go wrong as well. So we can now go over to Q&A.
Hi, thanks for that, Tim. Yeah, thank you for that information. It was really comprehensive on disinfectants. We've had a number of questions come in, which is unsurprising, and I'd like to ask you a few of those if we can in the remaining few minutes that we have. So I'm just going to pull them up. Okay, so the first question, hang on me one moment. So this is giving a scenario whereby in the case that a manufacturer of disinfectant is changed, would revalidation be required? Does that make sense?
Yes, so I think that's an interesting one. So presumably it's exactly the same disinfectant, but it's been undertaken by a different manufacturer.
Yeah, that's how it's reading.
Yeah, so I think it really depends on the formulation and the complexity of the disinfectant, and certainly change control and risk assessment will need to evaluate that. So if it's something simple like an alcohol, like ethanol or something like that, then probably not. If it's something complex like a quaternary ammonium compound, then it could well be subtly different in its chemistry. So I don't know if there's no right or wrong answer. Probably in most cases it would be prudent to do something, and even if it was just a field trial. So I'd lean towards doing something unless there was a good reason why not.
Okay, all right, thank you for that comprehensive answer. Right, the next question, this is in relation to biologic facilities. What are the best types of disinfectant to use in those types of environment?
Okay, so that's an interesting one. So is there a right type of disinfectant to use? The thing is that if I answer it going backwards, so for sporicides, the choices of sporicides is very limited in that balance between safety and efficacy. So really it's chlorine related compounds. So it's chlorine dioxide or hypochlorous acid, something like that. or it's a peroxygen like hydrogen peroxide or peracetic acid, or some kind of combination or blend between them. In terms of the sort of more general day-to-day disinfectant, then there's amphoterics, phenolics, quaternary ammonium compounds are sort of the main groupings. Probably, from my experience, quats are generally the kind of best overall performers, but every facility is different, and it sort of depends on the surface types and the bioburden. So I can only really answer that in very general terms, unfortunately. But it's
no, of course. Thank you for that. Just the audience has given some quite tough questions, so apologies for putting you on the spot. Okay, so the next question that's come in: are there limits that you can put in place if you perform surface conductivity for residue? What happens if you fall outside of those limits?
Okay, so there's two. I mean, there's two approaches. Some people would only look at a visual. So they'd say, can you see a residue or can't you see a residue? And you can get variations on different surfaces. So like residues on glass are quite easy to see, whereas residues on vinyl are less easy to see. In terms of conductivity, there are some published studies, and they do have recommendations for levels of conductivity that might indicate the presence of inorganic materials that are left over, but those values differ on different disinfectants, and you also need to take account of the surface material. So there is no like generic answer,
Okay.
but it's something again you can establish. So if you kind of do those measurements as part of the field trial, and then you do see reductions to relatively low levels, or you've showed that there's no need indeed to do any kind of residue removal, and you've got good data, then you could kind of put all that together to try and create the picture. So again, it's a really good specific question, but to which out of context I can only give a general answer.
Yeah, no, of course. Okay, I think we've got time for one last question. So again, this is sort of pertaining to conductivity. So the question is, if the use of conductivity isn't explicitly, oh, I can't get my words out, apologies. Okay, I'll start again. If the use of conductivity isn't explicitly stated in the guidance, why should sites use this testing that puts them at a disadvantage?
They don't have to. Okay. So what I said there was I put it out there as a potential. It's really that interaction of residues can cause one disinfectant to perform poorly, but you could go with low residue disinfectants. You could just do a rinse, and if you've got good data, then you've got good data. If you want to go the extra mile and make those assessments, or if you've got worries, then equally you can do. No one's saying you have to, but it is increasingly a challenge question from regulatory inspectors, particularly from MHRA, is how are you managing residues, and how you want to answer that will depend on where you want to position it. And if you have got conductivity data, it's much easier to answer than if you haven't, but there are different ways to answer that regulatory challenge.
Okay, thank you for that, Tim. I'm sort of mindful of time, so I just want to firstly thank you, Tim, for your time today. It's been really informative, and also to thank those who are attending the webinar that's been hosted today. I hope you've all found it very useful, and we appreciate the questions that you have asked. If we have any questions that we didn't cover during the Q&A, I will forward them on to Tim for his attention, and then we can circulate these along with the recordings at a later date. If you wish to catch any of our previous webinars or Tim's blog or regulatory updates, please go onto the RSSL website. We've got loads of resources that are really useful. And also we do lots of social media posts on the subject. Just touching back on RSL briefly, we offer a range of technical training courses which may be of interest either to yourselves directly or to your colleagues. So training courses are designed for professionals across the industry. We've got several upcoming sessions which you can possibly see on the screen, hopefully. So yeah, lots of helpful training courses there. So sterile product manufacturing, auditing against Annex 1. So yeah, if you want to contact me directly, then please feel free to do so. Those are my contact details, and I'll try and support you any which way I can. So just keep an eye on the website for future webinars, and I wish you all a very happy rest of the day. Thanks, Tim. Thanks, guys.
Thank you.
Thank you.